Welcome to the deep dive. Sometimes you're staring at a huge pile of complex information, papers, data, and you just wish you could zero in on what really matters. That's exactly what we're doing today. We are diving deep into the fascinating and honestly pretty mind-bending world of protein structure and folding. Now, these aren't just abstract molecules we're talking about. They are the absolute workhorses inside every cell, and their shape. Well, their 3D shape dictates everything they do. So our mission is to unpack this incredible hierarchy, how proteins achieve these precise forms. From the simplest starting point, that sequence of amino acids, all the way up to these intricate molecular machines. We'll dig into the forces at play and really try to understand why this molecular dance is so incredibly crucial. And we're drawing in some great material today, including insights from Lex 6 Cell Bio Podcast, levels of protein structure.pdf, and parts of Principles of Biochemistry.
And it's definitely a crucial topic. Understanding protein structure isn't just for biologists, It's fundamental to medicine, bioengineering, everything, really. It's essentially about figuring out how nature builds its most effective, most sophisticated tools.
Okay, so let's start right at the beginning there. The absolute foundation, that's the primary structure, right? Just that unique linear sequence of amino acids all linked together by strong peptide bonds, like beads on a string.
Exactly. A specific string of letters, you could say.
But here's where it gets really interesting and maybe a bit non-intuitive at first. This simple linear sequence, this string, it somehow determines the protein's entire final 3D folded shape. It contains all the instructions.
That's the central idea, often called Anfinson's dogma. The sequence holds the key.
It's like the DNA for its shape. And looking at evolution, we see that even when sequences change over time, certain parts, certain regions stay remarkably the same. They're conserved. And these conserved bits, they're not just there by chance, are they? They're critical, structurally, functionally. They're the parts the protein can't afford to lose.
Absolutely critical. And it makes sense, that if you have proteins with very similar sequences, they'll probably fold into similar shapes. That feels intuitive.
Right. Similar blueprints, similar building.
But here's the twist, and it's a big one. You can have proteins with quite different sequences that end up folding into the exact same overall 3D structure. Sometimes they even do similar jobs.
Wait, really? Different sequences, same structure?
Yeah.
How does that work if the sequence is the instruction manual?
Well, it implies that there aren't infinite ways for proteins to fold stably. There seems to be a limited set of preferred, energetically favorable architectures that evolution keeps landing on.
So nature finds the same solutions to different routes.
Precisely. And that's why classifying proteins based on their fold, their 3D shape, is so powerful. It cuts across sequence differences and helps us understand these underlying structural families and maybe even predict function.
So, for you listening, think about that. It means when we're trying to, say, design a new protein or understand how life evolved, it's not just the sequence. There are these fundamental principles of shape, of stability, that matter just as much. A different recipe might still bake the same kind of cake, functionally speaking.
A really powerful concept, yeah.
Okay, so we have the sequence. How does that 1D information start to become 3D? How does the chain begin to take shape locally?
Right. So we move from primary to secondary structure. And this is where weak interactions really come into play.
Weak interactions. So we're not talking about those strong peptide bonds anymore.
No, these are non-covalent. Think of them as more like subtle attractions rather than strong links. But there are lots of them. There are three main types we usually focus on. First, you've got van der Waals interactions. These are the weakest, very short-range attractions that happen between basically any two atoms that get close enough. They're everywhere, these tiny fleeting forces.
Like a general background stickiness?
Sort of, yeah. Then, a bit stronger, maybe 2 to 5 times stronger, are hydrogen bonds. These are more specific. They happen between polar groups, think water, or parts of the protein backbone, and they're directional. They like to line up just right. Super important.
Okay, H bonds, got it.
And the strongest of these non-covalent bunch are salt bridges, or ionic interactions. These occur between fully charged groups, positive and negative ions attracting each other. Depending on the surroundings, a salt bridge can be, maybe 10 times stronger than a van der Waals interaction.
Wow. Okay, so van der Waals, hydrogen bonds, salt bridges. And these create the first actual shapes.
Exactly. They drive the formation of regular repeating structures. The most famous is probably the alpha-helix. It's this elegant right-handed coil. The backbone atoms form hydrogen bonds, but not with their immediate neighbors. It's the N residue bonding with the N+4 residue 4 amino acids down the chain. That pulls it into the helical shape.
And the side chains, the bits that make each amino acid different.
Good question. They stick outwards from the helix. This is clever because it lets hydrophobic, water-hating side chains potentially tuck towards the protein's interior, while hydrophilic, water-loving ones can face the outside, interacting with the watery environment of the cell.
Makes sense. And the other main one.
The beta sheet. Here you have stretches of the polypeptide chain, called beta strands, lying side by side. They're linked by hydrogen bonds between the backbones of adjacent strands.
So less of a coil, more like flattened ribbons.
Yeah, often drawn as arrows pointing from the N terminus to the C terminus. They can run in the same direction, parallel or opposite directions, anti-parallel. Gives a kind of cleated structure, very strong. And then you have reverse turns and loops. These are the bits that connect the helices and strands. They're often on the surface, quite flexible, and don't underestimate them. They might look like simple connectors, but they can be crucial for function, sometimes forming the active sites where enzymes do their work.
It's amazing how these relatively weak forces acting together build such defined structures. But how does the protein find this correct fold? There must be gazillions of possible ways for a chain to fold up. Seems like it would take forever.
That's a classic question, the Leventhal paradox. And the answer seems to be cooperative folding. Proteins don't randomly try every single possibility. It's more like rolling downhill into a funnel. There are many unfolded, high-energy states at the top, but the landscape guides the protein rapidly towards the single, stable, low-energy native state at the bottom.
Driven by thermodynamics.
Absolutely. The hydrophobic effect is a huge driver. Nonpolar bits wanting to get away from water causes the protein to collapse inwards, and it's efficient. Folding isn't necessarily something that waits until the whole protein is made either. Often it starts happening while the protein is still being synthesized on the ribosome. Co-translational folding.
It starts folding before it's even finished being built.
Yeah, which helps narrow down the options early on. So the protein itself becomes more ordered, its entropy decreases, but the surrounding water molecules become more disordered, so the overall entropy of the universe increases. It's thermodynamically favorable.
Wow. Efficient and elegant. It's like self-assembling origami guided by physics.
You could definitely think of it that way.
Okay, so we've gone from the linear sequence primary structure to these local folds, the secondary structures like helices and sheets. What's the next level up? How do these pieces assemble into the final working protein?
Right now we're talking about the overall 3D architecture.
Although it's worth mentioning, not all proteins go through super complex folding, right? I'm thinking of things like fibrous proteins.
Exactly. Proteins like keratin, the stuff in your hair and nails, or collagen, which is incredibly abundant, kind of like the body's scaffolding. These are often dominated by their primary and secondary structures, forming long, extended fibers. They don't have much complex globular folding because their job is mainly structural strength or elasticity.
So less intricate folding, more about repeating patterns for strength.
Precisely. But for most proteins, especially enzymes and signaling molecules, we get into tertiary structure.
And that is?
That's the overall three-dimensional arrangement of all the secondary structure elements. The helices, sheets, loops within a single polypeptide chain, how that one chain folds up on itself completely.
Okay, one complete chain folded up, and then there's another level.
Yes, quaternary structure. This applies only when a functional protein is made up of more than one polypeptide chain, or subunit. Quaternary structure describes how these individual chains pack together. Think of hemoglobin carrying oxygen in your blood that's made of 4 separate globin chains interacting.
So tertiary is one chain's 3D shape. Quaternary is how multiple chains team up and what holds these higher structures together. Still those weak interactions.
Primarily, yes. Van der Waals forces, hydrogen bonds, salt bridges, they're all still crucial for stabilizing the tertiary and quaternary folds. They dictate how different parts of the chain, or different chains, nestle together.
But you said primarily. Is there anything else?
There is one important exception, a covalent one: disulfide bonds. These are special cross-links that can form between the sulfur atoms of 2 cysteine amino acid residues. They're much stronger than the non-covalent bonds.
Like little molecular staples.
Exactly like staples. They add extra rigidity and stability, especially important for proteins that exist outside the cell where conditions might be harsher. They really lock parts of the structure together.
Okay. And within this tertiary structure, is it all just one big folded blob, or are there like functional sections within it?
Excellent point. There's a hierarchy within the tertiary structure, too. We see recurring patterns. The smallest recognizable combinations of secondary structures are called motifs, like a beta strand, then an alpha helix, then another beta strand, a beta-alpha-beta motif. These are common building blocks found in many different proteins. Even if the sequences are different, they just represent stable ways to connect secondary elements.
Okay, motifs are small structural patterns.
And motifs often assemble into larger, more substantial, compact units called domains. Now, domains are really interesting because they're often independent folding units, meaning you could, theoretically, snip out a domain from the rest of the protein, and it would likely fold up into its correct 3D shape all by itself. It maintains its own structural integrity. Most larger proteins are actually modular, built from several different domains connected together. Each domain might have a specific role. Maybe one binds a target molecule, another performs a chemical reaction. They work together.
Wow, that's incredible modularity. So for you listening, picture that. Nature isn't always reinventing the wheel. It's using these prefabricated, independently folding modules, domains, and combining them in different ways to create a vast diversity of protein functions, like using standard components to build complex machines.
It's a very efficient evolutionary strategy.
And just like that, we've traveled quite a path, haven't we? From that simple primary sequence, that list of amino acids holding the secret code, through the emergence of elegant local shapes like helices and sheets in secondary structure, driven by all those subtle weak forces and that amazing cooperative folding, all the way to the complex functional 3D architectures of tertiary and quaternary structure, stabilized by weak interactions and sometimes those disulfide staples and built with those modular domains. It really is a testament to the power and precision of molecular interactions. And understand this whole folding process is just so fundamental to understanding life, from how strong your hair is, thanks to keratin, to the enzymes running every reaction in your body.
Absolutely. It underpins almost everything. And maybe, to leave you with something to think about, consider this. We know the primary sequence dictates the fold, yet we also know different sequences can lead to the same fold. And these proteins are built from modular, independently folding domains. So what does this combination of determinism, flexibility, and modularity tell us about the basic rules of molecular design that life uses? And perhaps more practically, how could we potentially leverage this resilience and modularity? Maybe in designing new drugs that target specific folds, or in synthetic biology, creating proteins with novel functions, or even figuring out how to intervene when proteins misfold, which is the cause of many diseases. It feels like a huge frontier, still with so much to explore and harness.